All right. First, a little bit of course matters. Sort of the most current version of this. Sorry. Here we go. And we can do that whole screen. There we go. A reminder that Susan's going to be leading a review session tonight at here, 6 to 8. And I believe you're going to make some of the materials available later. Okay, so they're available on Icon, and they will also be here in a physical, incarnated form tonight. She's needed to reschedule her office hours because of Research Week, but she will hold them Wednesday, 2 to 3 at the usual place. Again, I can't say this enough. We're not going to be here Wednesday. We're going to be in Met Alumni. But we have an intrepid scout who is willing to go and find how to get there. And I will mail this to you as well, but I want you to hear it too. If you go to the Compass Rose in the General Hospital and go east to elevator BE, go up to the 3rd floor and take a left, you're there. And I will email that as well. I have a few copies of the map if you did not receive them in email. Everybody ought to have received a copy of the map by email, but if you did not, there are copies there, and it means you're not receiving anything else from the course, which should concern you. And then Friday will be the exam. Chapters 11 through 15. Material today could appear on the test. Do people have questions about any of those just sort of practical housekeeping type things? Yes? The exam is here. The shift on Wednesday is a one-day only disruption to our lives. Anything else? Other practical questions? There should be another practice exam. If it's not up yet, let Brenda know and she'll put one up. Any other practical questions before we can get back to the fun of science? Okay. So I'll close that. And I will close that. I thought I would just show you that map one more time. Oh, well. You've got it. Okay. There we go. Now, I believe that we had gone in our handouts about this all the way through the amino acids as neurotransmitters. Is that correct? And we should therefore be about to start purine metabolism, nucleotide biosynthesis. Is that about right? Or are we further along? This is when? We've got a little farther than that. Not much. No, this is about it. This is about right. Nucleotides have to be synthesized because we have to have DNA and RNA. Even though their synthesis can be quite complex at times, the only organisms that don't have nucleotide synthesis are things that are parasites, generally intracellular parasites. But a free-living organism will always have a way to make nucleotides. And obviously, since the nucleotides have a lot of nitrogen in them, you're not probably surprised to find out that the amino acids are one of the major precursors for making them. Now, the fact that we can make them doesn't mean we sneer at the ones we get for free. So if there are nucleotides in the diet, they will be taken up and they will be used gratefully. In addition, our body has salvage pathways so that after nucleic acids are broken down, we have ways to capture that and recycle them in a very thrifty way. So the purines, those are the big ones, AMP and GMP, share the fact that they're built on this PRPP base. And I know we met that before because we met that in histidine synthesis. And it has a long name, but a good one. So it's pyrophosphate. We're going to have phosphoribosyl pyrophosphate. So it's a phosphoribosyl here that has a pyrophosphate hanging off of it. So this synthesis, taking ribose 5-phosphate and making a PRPP out of it, is the first step for making all the purines. And so this enzyme, ribose phosphate pyrophosphokinase, it's letting you know it's putting on 2 phosphates at once. is an initial step, and obviously an expensive one. It'll cost us 2 high energy bonds right off the bat before we get any further along. Purines take 10 steps to synthesize. I'm not going to go through all those 10 steps. There are at least 5 different sources of things, and then we'll take a little bit of a look at that. If you look here at the inosine monophosphate, which is the common precursor to both AMP and GMP, we can take a little look at some of the places that stuff has come from. So here we've incorporated an entire glycine. We stole just the nitrogen from an aspartate. Here's folate giving us a carbon. Here's nitrogens from glutamine. Glutamine's a good nitrogen donor for a lot of purposes. Here's another carbon from folate, tetrahydrofolate. And here's a carbon and oxygen that just came from bicarbonate. So it's a real hodgepodge. It's like your grandmother's quilt. In general, the nitrogens are coming from amino acids. So that means that amino acid metabolism and nucleotide metabolism are intimately and irrevocably connected. The carbon backbones come from a variety of places, so they're going to come from amino acids, plain old bicarbonate, and tetrahydrofolate. And this is one of the reasons why folic acid is so important in growing stages and or any time that requires growth. If you have to have tetrahydrofolate to make nucleotides, then if you don't have tetrahydrofolate, you're likely to have problems. And that is in some way or another behind the observation that spina bifida can be prevented by adequate folic acid in the diet. Now, here is where it diverges. So you have inosine monophosphate. The phosphate acts as sort of an activating tail. That's all it needs. And we want to arrive ultimately at both AMP and GMP. In the case of AMP, the next step is to actually just add an entire aspartate molecule through its nitrogen. So the nitrogen here has replaced the oxygen, the carbonyl oxygen there. So we put on an entire aspartate for the moment. In a sense, the carbon backbone of the aspartate acts as the activating group. We can then split off just the carbon part, the 4-carbon backbone, and that's fumarate. And I know you all recognize fumarate as a citric acid cycle intermediate. So now we've gotten to AMP, one step. It's powered by GTP. Okay? The other pathway, if we want to go from inosine monophosphate, we're going to have to invest some reducing power, which oddly enough is NADH in this case, or oxidizing power. We're going to oxidize it. And so in addition to the oxygen up there, we're going to have an oxygen down here because we've oxidized it. And that's xanthocene monophosphate. Xanthine is a waste molecule in certain other processes. And then that carbonyl oxygen is set up for another nitrogen transfer, in this case from glutamine. And so that nitrogen here came from the amide tail of glutamine because it left glutamate and was expensive. It was fed by ATP. And it cost us 2 high energy bonds. And so, since each fork of the path depends on the opposite product, it's self-regulating. Since you have to have ATP to make GMP, you're not going to get way ahead on GMP. Since you have to have GTP to make AMP, you can't get way ahead on ATP. It's a self-regulating, self-balancing setup. Very, very sneaky. And both arms of it depend on amino acids, and so that helps to knit together all the nitrogen pathways, and it also helps to couple nucleotide synthesis to nutritional status, whether or not you're adequately fed. Any questions so far? Okay. Now, in the case of pyrimidine biosynthesis, we start at the other end. We're going to make the base first. We're going to make the ring structure, and then attach that to PRPP. And if we take a look at the ring structure, here we have, again, grandmother's quilt. We've incorporated basically all of aspartate here. We've incorporated just a nitrogen from glutamine, so glutamine's playing a role again And bicarbonate has again offered us a carbon and an oxygen. And then once that base has been assembled from different components, then it's attached to the PRPP base, the phosphoribosyl pyrophosphate. So PRPP is common to both purine synthesis and pyrimidine biosynthesis. And again, it's taking advantage of amino acids for a lot of its components. Making UMP is inhibited by any of the derivatives, UMP, UTP, UDP, all of those things will inhibit its synthesis. But it can be activated by ATP. So if there's purines to spare, maybe it's time to make some pyrimidines to make up for that and get back in balance. So CTP, we still need CTP. It goes from UTP. Here's the UTP molecule with its 2 oxygens hanging off. And just as we saw before, glutamine is going to donate its amide nitrogen at the expense of an ATP. And so this carbonyl oxygen turns into an amine group here. And now we have CTP. Again, it costs us a bit of energy. Again, it involves amino acids. Now, all that we've made so far are ribonucleotides. And ribonucleotides are obviously only useful for making ribonucleic acids. But, at least at our level of life, ribonucleic acids are not enough. We need deoxyribonucleic acids as well. And so somehow or other, we've got to take the ribonucleotides and make deoxyribonucleotides. And so here is the ring of the ribonucleotide. It's got its 2 oxygens hanging off. And I think we're going to skip this mechanism. I think we've been doing a lot of mechanisms, and I think we have more to go. So I'm just going to skip that for you. You're welcome to read it if you like. This leaves us only one nucleotide left to worry about, which is phenidylate, TMP, TTP, so forth. Here we have deoxy-UMP, fondly called DUMP. So the ribose ring is the deoxy form now. You recognize the UMP. And we need to make TMP. We need to add a methyl group there. That's the only difference between the uridine and the thymidine is one extra methyl group. So it really can participate in all the same hydrogen bonding activities. The methyl group just acts like a bump to let you know that it's there. And this is again taking a carbon from tetrahydrofolate, this carbon donor that's played a role so many times before. And the enzyme to do this is called phenidylate synthase. And phenidylate synthase is an enzyme that can actually do 2 activities in the synthesis of making deoxy thymidine monophosphate, DTMP. And it starts on one end of the enzyme and is at one end of the enzyme and then scooches across the enzyme to the second active site. This blue area here is an electrostatic surface that acts like the sticky stuff on Post-it notes to keep the intermediate sort of on the enzyme sliding around. And when it gets to the active site, it's ready for the last reaction. So phenidylate synthase is specifically an enzyme involved in the nucleotides needed for making DNA. Most of the enzymes we've talked about so far would be equally important for either RNA synthesis or DNA synthesis, but since thymidine only occurs in DNA, this enzyme is specifically important in the synthesis of DNA. And that's why the drug methotrexate, you may have heard of methotrexate, it can bind to this enzyme and mimic tetrahydrofolate. And so it acts as an anti-proliferative agent. It can prevent cells from growing because they can't make more DNA. They're not necessarily going to die right off, but they can't grow, they can't multiply. And it's used as an anti-cancer agent. It's used as an anti-arthritis agent and other instances as well where one kind of cell or another has gotten way over its depth. So, so far we've been talking about the synthetic side of nitrogen metabolism, building things up, the anabolic side. You've done enough metabolism now to know that where there's anabolism, there's always catabolism. There's always going to be some way to break it down as well. Amino acids make perfectly adequate fuels. They have energy in them, just as sugar does. And they're especially the major fuel for the small intestine. You know, that's going to be the part of the gut that absorbs the interesting parts of food, things that are nutritional. What gets past the small intestine is passed up to the liver through the circulation, and it can also use that as fuel, or it can use the backbones of the amino acids to make glucose or ketone bodies. And you know how it would use it to make glucose through gluconeogenesis, and you know how it could use it to make ketone bodies through acetyl-CoA. The body never stops making some sugar. It always is going to make some sugar. How much will depend on how long it's been since last eating. And if you have not been eating food, those amino acids have to come from internally. There's not a very large pool of amino acids in the body. And so at that point, it's your own muscles breaking down that release amino acids for the liver to do this. When we look at the different amino acids, we can divide them into two kinds of amino acids in terms of what they can be used for as fuels. And we call them either glucogenic, because their carbon can end up in glucose, or ketogenic, because their carbon backbones can end up in ketone bodies, and a very few are going to be both. And let's see how that looks, just sort of, you know, some examples of that. So here we have asparagine. It's a 4-carbon backbone, has the amide terminal bit there. It's got a free amine group there. The first thing that's going to happen is an enzyme called asparaginase is going to just throw away that ammonia. Okay, just cuts it off as an ammonia. And now we have aspartate. And this nitrogen can be stripped off through a transaminase reaction, and alpha-ketoglutarate can pick it up as glutamate. And now we have oxaloacetate. Oxaloacetate is obviously equally good as a citric acid cycle intermediate or as a carbon backbone for gluconeogenesis. This exact same logic can be used on the 5-carbon amino acids. So glutamine can have its ammonia thrown away. Glutamate can have its nitrogen, its ammonia picked off, and that would leave you with alpha-ketoglutarate. So these amino acids and these Krebs cycle intermediates are really intimately related. And we saw when we were studying the citric acid cycle, them being used to synthesize amino acids. This is how they can be used to fuel. A couple of shorter amino acids here, serine. It's a 3-carbon backbone. You might think if it's a 3-carbon backbone, it ends up making pyruvate, and it does. So all you have to do is get rid of the ammonia, and you actually have, well, not quite that simple. You probably have to take care of the alcohol. But you end up with pyruvate. Cysteine is also a 3-carbon backbone. In this case, you're going to have to get rid of the sulfur as well. And to fit these two on one slide, I did some mirror writing here, so forgive me. And the nitrogen is just thrown away in each case as ammonia. Sorry. And the sulfur is lost as hydrogen sulfide. So you're throwing away valuable components here. If you remember how much energy we put into picking those things up, we're throwing away energy when we throw them off. So we've seen here how many of the sort of simpler amino acids are glucogenic precursors. So we've got serine and cysteine with its 3 carbons to pyruvate, aspartate and asparagine directly to oxaloacetate. Pyruvate could be carboxylated to oxaloacetate. Glutamine, glutamate through alpha-ketoglutarate. So we've already accounted for 1, 2, 3, 4, 5. We've accounted for 6 of the amino acids already as either intermediates for the citric acid cycle itself or for gluconeogenesis. threonine's kind of funny. It actually does both. Threonine is a 4-carbon amino acid, but it's a branched chain, if you like, because the alcohol is there and the methyl group is off there, so it's sort of a branched amino acid. And some of that energy can be picked off in NAD becoming NADH. So now that alcohol is a carbonyl oxygen here, a keto group. CoA can come in and split that in the middle, releasing a glycine molecule and an acetyl-CoA. So a little bit of energy is coming out at the beta carbon when the NAD plus is oxidized. So this is able to go into both ways. Glycine is so tiny, you sort of wonder what to do with it. We've mentioned before that glycine can be a precursor to serine, and that's one way it can go, is it go off to serine and then to pyruvate. The principal pathway we see is this one, that tetrahydrofolate can cleave this into a useful carbon and throw away a carbon dioxide. So the central carbon here is going to get plucked off by the tetrahydrofolate. The nitrogen is lost as ammonia. The carboxylic acid is lost as carbon dioxide. But we do manage to reduce an NAD plus in the process. So we've scavenged a carbon that's useful at the expense of losing ammonia and carbon dioxide. But tetrahydrofolate's a good thing to recharge, so it's not all for vain. Valine is a little more difficult. Here you can take a look and see where we're going to be heading. Valine is again a 1, 2, 3, 4, 5 carbon backbone, but it's a branched backbone. And if you remember, branched fatty acids were an issue back when you're doing lipid metabolism. And so what are we going to do? Well, we can get rid of the nitrogen here by giving it up to alpha-ketoglutarate. And now we have, by transamination, and now we have a carboxylic acid. And this is a branched chain alpha-keto acid. And we can pick it up with CoA. CoA can be the carrier molecule for it. And in the process give some NADH, which is always good for energy. We're going to pay for it with a carbon dioxide lost. We have then a branched chain acyl-CoA. This molecule can be further oxidized with electrons ending up in QH2, and the QH2 obviously would be coming in at complex 2 in the electron transport chain. And this would be the same enzyme that fatty acid oxidation uses. And from there we can end up making succinyl-CoA. So let me see if I can test that. There's a disease that can occur if you don't have the enzymes to do this. This dehydrogenase is key to it, and if you don't have it, certain metabolites accumulate, and the disease is called maple syrup urine disease. And it was observed by a Norwegian mother who had 2 boys whose urine smelled funny, and she was concerned about them. And it was her bringing it to the attention of pediatricians that uncovered this as a deficiency disease. The aromatic amino acids are also going to be a little interesting to try to degrade because they're kind of bulky and odd. They are also glucogenic and ketogenic, like threonine. Their products are going to be acetoacetate, either alanine or fumarate. And here we can start, for instance, with phenylalanine. And phenylalanine, of course, is just alanine with a phenyl group on it. And that itself is not something we can degrade, but we can activate that by making tyrosine out of it, which we might do if we wanted more tyrosine. So this enzyme is sort of a bifunctional enzyme. Phenylalanine hydroxylase is both useful for synthesis and for degradation. And it makes tyrosine out of it. This is the enzyme that's missing in phenylketonuria. That if you can't turn phenylalanine into tyrosine, phenylketones accumulate in the urine. And as you probably know, that has to be dealt with very shortly after birth. The infant has to be put on a very strict low phenylalanine diet. And why would it only matter after birth? Well, what's wrong? What spares the baby up until that point? Exactly. The metabolic wastes are transferred to the mother, and then the mother can pass them out through the kidneys. But if you actually want to get rid of it, if you want to throw it away, or if you want to burn it for energy, then a transaminase will give you parahydroxy. There's the hydroxy part, and it's on the other side, so it's parahydroxyphenylpyruvate. So this is the phenyl ring. There's our friend pyruvate. It's like Zelig. It's everywhere. I hope that movie isn't too old to be Anyhow, so we've got paraphenylpyruvate. We're going to oxidize this ring further. That's still kind of a resistant ring. And it's a dioxygenase. You can see it's oxidized on 2 carbons to homogentistate. And then that is further oxidized to mallyl acetylacetate. So now it's one long chain, funny looking carbon backbone. You can figure out that it must have been split either there or there. This enzyme is missing in alkaptonuria, and in the absence of that enzyme, the urine turns black after it stands for a while, that the homogentisate oxidizes in the air. But I think you can see why this would be both potentially ketogenic and glucogenic, why you could get those 2 kinds of backbones out of it. Well, we've been splitting off ammonia with wild abandon and not worrying about it. But we have to do something with ammonia. You can't accumulate ammonia indefinitely because it's an alkaline compound. It's going to raise the pH of things. It's rather toxic to the brain, so that in kidney failure, dementia due to ammonia accumulation is sometimes one of the first symptoms. And because it's abundant, it can end up killing the citric acid cycle because you withdraw the carbon backbones to make amino acids, even though you don't need them. So we've got to get rid of that ammonia. There's this compound, urea, and as you can see, it can carry 2 nitrogens for each carbon. So it's a very efficient carbon carrier. It's also a neutral one. There's no charge on it. It's also quite polar, so it's very, very, very soluble. You can get up to what % urea? Something phenomenal. Very, very. You can get 8 molar urea into solution. So urea is practically a solvent. This was the first synthetic organic compound made entirely from inorganic materials. One of the first nails in the coffin of vitalism. So we get rid of about the urea accounts for about 80% then of the nitrogen. So how does nitrogen get to the urea? Well, we've been accumulating ammonias onto glutamate, if you remember. We would let it come in and carry off a nitrogen for us. And I don't know why the formatting has done this to me. So we can have here glutamate, and this is very irritating. Anyhow, what we want to end up with is alpha-ketoglutarate. So we're going to end up having a way to take off that ammonia. But this has to be oxidized in order to get it off. And this is actually able to use either NAD plus or NADP plus. It's very unusual to be able to use either one. And I think I have a typo here. It should say NADP plus or NAD plus. Obviously, you can't use both the oxidized and the reduced form. So there's a typo in there. You could correct that. This should be NADP plus. That should just be NAD plus. Forget the pH. These reactions are reversible and can be used in either reaction. So you can think of it either as a catabolic pathway to get rid of excess nitrogen, or as an anabolic pathway to fix nitrogen that's available. So this is really an anabolic reaction. But we need to somehow get that ammonia into urea. The ammonia is toxic. The urea is essentially non-toxic and very soluble and neutral. So we've got to get the ammonia, which is a completely reduced compound, into urea, which is a partially oxidized. And it's kind of a funny pathway to get there. It doesn't look obvious to begin with. We need to make an activated carbon backbone. And so we know we're only going to have one carbon left at the end. We can activate a bicarbonate using ATP and make carbonyl phosphate here. So this is a carbonyl phosphate. And this then is a substrate to pick up the ammonia, which can give us carbamate. And ATP can come in again and give us carbamyl phosphate. So here we have a good chunk of urea made up. This step put together the carbon, and then this step, in this step we added in the nitrogen. So now we have an activated form of 2/3 of urea. And then we get to the urea cycle. Why don't we start with a compound that we just made? So we've just made some carbamyl phosphate. Here we have the carbamyl phosphate. It's got a nitrogen from an ammonia and a phosphate from the ATP. It can then be added as a chunk onto the end of ornithine. Look at ornithine, 1, 2, 3, 4, 5 carbons with an ammonia there. It's an analog of lysine. It's not lysine, it's an analog of lysine, and it can be the recipient for the carbamyl phosphate. And because it has the phosphate tail, the energy is there to make citrulline. Still a 5-carbon backbone, and now with this growing, interesting end down there. It was first isolated from watermelons, which is why it's called citrulline. Citrulline is going to be the target of an attack by aspartate. Here is the amino acid aspartate, and at the cost of ATP, that nitrogen is going to get smushed on to the terminal carbon, and now we have argininosuccinate. So this part here, like that, is arginine, the amino acid arginine, with a succinate hanging off of it. And we've actually seen something like that happen during nucleotide synthesis, when we put on an entire aspartate and then took off a citric acid cycle intermediate. So here's argininosuccinate, and when we split it off, it comes off as fumarate. It leaves us an arginine molecule, and the arginine can then have the terminal guanidino group down here removed by hydrolysis as urea. Here we've got the urea we were trying to get. One nitrogen came in as carbamyl phosphate. Second nitrogen came in from the aspartate. And now we have our urea molecule, and we've regenerated our ornithine. So this can either be looked at as a degradative cycle that makes urea, or it can be looked at as a way to make arginine if you can refeed the pathway with ornithine. This is some of the reactions through which we make arginine when we're young and have no desire to get rid of nitrogen, for the most part. Just looking at those reactions in a little more detail. Carbamyl phosphate, the carbon here from the bicarbonate, that came from the ammonia. The lysine analog, smush it onto the end, and we have the citrulline. Let me just take a look. Okay. So this is still a 5-carbon backbone. And argininosuccinate is going to be, the aspartate is going to be attacking through the nitrogen. It's going to attack that carbonyl that keto group there, and you end up with a nascent guanidino group. This would be arginine like this, and that would be succinate. So this is going to be an expensive reaction, cost us 2 high energy bonds to do that. Making urea to throw away costs a lot of energy. Argininosuccinase is the enzyme that can then split that off, and one part is going to come off as fumarate and the other as arginine. So we have the original nitrogen from the carbamyl phosphate and the second nitrogen from the aspartate. The fumarate can go back and potentially be made into another aspartate. Here we have the arginine, and arginase can just simply hydrolyze here. That's a bond that's relatively easy to break, so just hydrolyzes the bond there, and when that chunk comes off, it comes off as the molecule urea. So this oxygen is coming from the water molecule that hydrolyzed it. And now we have ornithine, and we can go around again. It should be obvious that there is now an intimate connection between, the citric acid cycle and the urea cycle. Hans Krebs discovered both of them, and for many years the citric acid cycle was called the Krebs cycle. And some people like to jokingly call the two cycles together, the citric acid cycle and the urea cycle, Krebs bicycle. Biochemists have a strange sense of humor. Anyhow. And so we've talked about the pathways through which, by transaminases and glutamate dehydrogenases, we can get ammonias for the carbamyl phosphates. And then from these Krebs cycle intermediates, or citric acid cycle intermediates, fumarate, malate, oxaloacetate, we can make the aspartate to feed through round and around. So really there's an intimate relationship between the citric acid cycle and the urea cycle, between the two pathways. Mm-hmm. How does something like that get controlled? Well, if you have such a rich diet that you are breaking down amino acids, or if you have to break down amino acids as a fuel, it will lead to high levels of glutamate. And that in turn will increase the synthesis of N-acetylglutamate. Here we have glutamate by itself, and here is acetyl-CoA, which might come, for instance, from fatty acid oxidation. And this molecule, N-acetylglutamate, is an allosteric effector of the carbamyl phosphate synthetase. It will increase the rate at which carbamyl phosphate is made. And if there's more carbamyl phosphate made, it increases the rate at which the urea cycle is going to go around. Urea is not the only way of getting rid of nitrogens. It's one that we've come up with given our kind of intermediate water status, if you like. Birds and reptiles both live a lifestyle where they can't use up a lot of water. They're such high metabolic rates that it would take a lot of water to satisfy them, or they're living in places where there just isn't a lot of water, whatever. There's a trick that they use, which is to make something that precipitates as a solid. So you remember, I hope, xanthine back from our discussions of purine metabolism, that xanthine was one of the, xanthine monophosphate was one of the intermediates. Here's just plain old xanthine. And you can see that xanthine is very nitrogen rich, so there's 4 nitrogens in that backbone. And if you oxidize this, you can make it into uric acid, and it will become substantially less water soluble and precipitate out as what you see on top of your car if you park under the tree near the campus cinema. We also can get uric acid from nucleotide degradation. So if you have a diet that's rich in nucleotides, you can make a lot of uric acid. Some of the things that are rich in nucleotides are things that are rich in young cells. So mushrooms, broccoli, I think asparagus may be rich in it, red meat to some extent. And in this case, the uric acid is in the circulation and can deposit in the joints. So it's the depositing of uric acid crystals that cause gout when they form in the joints and cause great pain, or they can precipitate in the kidney and make kidney stones. So a good reason to drink a lot of water. All right. So what's a waste for one person can be a raw material for another. Urea is the common form to excrete nitrogen for many animals, but it's useful nitrogen. It's nitrogen that has already been fixed into an organic form, and so it shouldn't surprise us that there are organisms that take advantage of that. And so both fungi and bacteria can take urea, which is by itself unscented, and the enzyme urease to hydrolyze this molecule into 2 molecules of ammonia and a molecule of CO2. And then these organisms would just take that ammonia and fix it by the pathways we've already discussed for fixing nitrogen. Interesting thing about urease is that it was the first protein to be crystallized. And this was revolutionary because it was proof that proteins were discrete, coherent things. They weren't colloids. They weren't nitrogenous colloids. They were a thing. And urease was different from, another one. And of course, it was necessary to get crystals before their structure could be determined. Fish swim in water, so water is not a problem for them, and they just simply let the ammonia float away through their gills. And that's it. I'd be happy to answer any questions that people might have. We're going to be in Med Alumni. You'll get a good lecture here, but it won't be me.